In this video, I'm going to briefly introduce some of the bioinformatics software that we will be using for this DNA barcoding project. One of the main ideas behind DNA barcoding is that it can be used as a tool for rapid species identification. The idea is to use a sequence that is common to all animals or plants that can, 1, contain specific species-level genetic variability, and 2, possess conserved flanking sites for developing PCR primers, and 3, the sequence has to be short enough so that it can facilitate current capabilities of DNA extraction and amplification. Cytochrome C oxidase in animals and MATK and RBCLA for plants are the sequences used for DNA barcoding. So this 400 to 800 base pair fragment will serve as a unique fingerprint for species. So after you've collected your specimen, you can then collect a tissue sample, extract the DNA, amplify it, and then sequence it. Following that, you might want to then apply some bioinformatics to clean your data, assemble your data, identify the species related to the sequence data, and then possibly perform some molecular phylogenetics analysis. So basically everything at this yellow arrow here is what I'm going to talk about or share with you today. So what is bioinformatics? This is an interdisciplinary field that creates and uses software and tools to store, manage, and analyze huge collections of biological data. The ultimate goal is to help biologists better understand what is going on inside the cell. So for example, after sequencing an entire genome, bioinformatics applications can be used to predict whether proteins and RNA encoding genes are located on the chromosome or chromosomes. We can predict the functions of these proteins and RNA sequences. We can perform genomic comparisons, where we will compare a newly sequenced genome to a more well-studied organism to identify novel genes or missing genes. We can even go a little further to predict what biological pathways may exist in this new organism and how it might be affected with these new or deleted genes. DNA sequencing is a procedure that aims to identify the exact order of every nucleotide on a strain of DNA. So currently, Sanger sequencing technology can only identify base pair fragments that span between 800 and 1,000 base pairs, which works fine for the purposes of DNA barcoding. So each fragment is then assembled into a larger contiguous string of characters until the complete chromosome is then has been sequenced. And one of the first things you'll encounter is what's called a DNA trace file. The DNA trace of a read shows the results of a sequencing run. So we can use tools such as FinchTV to clean the data. And this is a snapshot of what FinchTV looks like. Cleaning the data involves removing poorly identified nucleotides at either end of the read, as well as maybe completely throwing away data if the trace shows a lot of noise or poorly defined peaks for each character that represents a specific nucleotide. Once the reads have been cleaned with software such as FinchTV, we can now assemble them. In DNA barcoding, you'll receive a forward and a reverse strand that overlap. We would like to use the assembly of these two sequences. So bioedit, here's a snapshot of bioedit. Bioedit is a bioinformatics package that provides an assembly tool called CAP. And CAP is short for Contig Assembly Program. This tool can be used to assemble two DNA sequences that you'll play with later, and it'll assemble them into one contiguous sequence. That is, if it can be assembled. Next, we would move to a program called BLAST, which is short for Basic Local Alignment Search Tool. This is a sequence similarity search tool. So you give it a query sequence such as a DNA or protein sequence. It will then compare that sequence to a database of either DNA or protein sequences. And with this comparison, it'll retrieve for you any sequences that are significantly similar. And this is particularly useful when we want to identify the species name of our specimen. So in this figure, I submitted a DNA fragment into BLASTX. So BLASTX translated the sequence and then identified the protein sequence from its database that most closely resembled my sequence to help me identify the specimen. So there are multiple sequence alignments. And multiple sequence alignment is an alignment between 3 or more sequences. The goal of the algorithm is to position all the sequences with respect to each other such that it maximizes the number of matches in all the vertical columns. It wants to maximize identical matches and maximize the number of conservative substitutions while at the same time minimizing the number of mismatches in vertical columns. These types of alignments are useful in a molecular evolutionary analysis because it enables you to identify regions within that family, that gene family or that protein family, that are resistant to mutations, and other regions that are accepting of mutations. Now, this data is particularly important as the input or for the input for molecular phylogenetics analysis. Now, molecular phylogenetics is used to infer evolutionary relationships between species using molecular data from those organisms. So, for example, if we were to use fragments of cytochrome C oxidase in a molecular phylogenetics analysis, it would tell us specifically that the translation of the DNA fragment of cytochrome C oxidase from, for example, spider N here is more closely related to the translation of the DNA fragment of cytochrome C oxidase from Goldweaver than to any of the other sequences in our data set. We then take this information to suggest or infer that these two species are more closely related to each other than to any of the other sequences within our data set. There are 5 analysis methods commonly found or that commonly come with molecular phylogenetic software. Here is a snapshot of software that I use called MEGA, which is short for Molecular Evolutionary Genetics Analysis. One of the more common analysis procedures is called neighbor joining, and the other one that you may find commonly listed in papers is called maximum likelihood. Now, with phylogenetic analysis, there's an inverse relationship between speed and accuracy. For example, neighbor joining is faster than maximum likelihood. However, it's less accurate. Whereas maximum likelihood is takes a lot longer to finish, but it is more accurate than neighbor joining. So if you have a lot of data and you want to quickly get an idea of the general evolutionary relationships in your data, then you would want to use neighbor joining. However, if you want a more accurate depiction of the evolutionary relationships, then you do want to use maximum likelihood. So I just want to briefly talk about some of the features in a tree before I end this presentation. So at the ends of the trees, you find the names of the sequences, and they are referred to as taxa. Each horizontal line is called a branch and has a length associated with it, and this length represents the amount of divergence for that lineage in the data set. The vertical bars have no meaning other than to serve the purpose of separating the horizontal branches far enough so that we can better see or read the taxa. The scale bar at the bottom indicates the amount of change per branch length. So the shorter the branch length, the shorter the amount of divergence. And the node circled in green represents a hypothetical ancestral sequence from which the taxa have been derived based on the sequences within our data set. Lastly, the numbers at each node are called bootstrap values. These values are confidence values or support values for each node. So with the bootstrapping procedure, several hundred trees are generated by randomly sampling the input multiple sequence alignment. These trees are then compared to the original tree to identify how often the nodes in the original tree were conserved in all of the generated trees. So for example, the node that bifurcates into spider N and Goldweaver here was conserved 100% of the time, or was conserved in 100% of the trees that were generated. However, the node that splits into caddy N and moth was only conserved 48% of the times within the trees that were generated from the bootstrapping procedure. This means that our data in the multiple sequence alignment strongly supports the relationship between spider N and Goldweaver, but not so much for the relationship between caddy N and moth. And that concludes my quick presentation on DNA barcoding and bioinformatics software.